really appreciate everyone coming here today, both online and in person. Very excited to share with you some great data that we just announced at EASL, and also give a company update on Genomics. We will have a Q&A later, so feel free to ask questions. Okay, so my name is Justin Lee. I'm the CEO of Genomics, an AI-based multi-omic test developer for the detection of early stage cancer. With me, Gary Frazier, our Chief Growth Officer, Alex Brenner, our Chief Financial Officer, and then on Zoom, we have Dr. David Taggart, our Chief Scientific Officer, Dr. Shivani Mahajan, our Head of Computational Science, Dr. Rick Van Etten, our Chief Medical Officer, and Dr. Mindie Nguyen, our Key Opinion Leader and Principal Investigator in CLiMB. We look forward to presenting a brief company overview today, including an update from Dr. Mindie Nguyen on our groundbreaking clinical trial results, demonstrating the performance of HelioLiver in a large-scale prospective trial. So by way of a brief introduction of the company, we were founded in 2016. Our original intellectual property was from UC San Diego, out of the Genomics Institute. We spun out 9 foundational patent families into Helio. These inventions cover our AI biomarker discovery platform engine, our methods for measuring methylated cfDNA, and cover specific CpG sites that are predictive of a multitude of different cancers. We are a multi-omics company, but first and foremost, we are a methylation company. We were one of the first pioneers in blood-based cfDNA methylation detection and techniques. We developed our first product, HelioLiver, for the detection of early-stage hepatocellular carcinoma, the most common form of primary liver cancer, or HCC, to address a severe unmet need with ultrasound, which is the current standard of care. It's also been a pain point for physicians, patients, and insurance companies for many decades. In 2021, HelioLiver received a CPT PLA code by the American Medical Association. In 2022, we received Medicare pricing for $662.32 on the clinical lab fee schedule, or CLFS. And after 5 years of running our pivotal clinical trial, CLiMB, we have finally unveiled data, most recently at EASL in Milan, the largest international liver disease conference. CLiMB was the largest of its kind. It was a fully prospective study, and we're very excited to share data from that, where we passed our pre-specified statistical endpoints, and we've gotten significantly positive feedback from the scientific and medical community. In May, we submitted our data formally to the FDA for PMA authorization as a Class 3 medical device. So you might ask, why liver? There are many reasons why we decided to tackle the liver market first. Amongst them, existing technologies are extremely inadequate. Currently, ultrasound is the gold standard. Ultrasound is known for having extremely poor sensitivity for detecting early-stage HCC. MRI is prohibitively expensive and only used as a reflex diagnosis test. The current recommendation by all major liver societies is ultrasound for surveillance every 6 months in high-risk individuals, and then reflexing to an MRI when ultrasound is positive. As a result, more than half of liver cancers are detected in late stage, and prognosis is very poor. Another major problem with ultrasound is that it is very inconvenient. Most clinics don't have the capability to perform ultrasound in-house and must refer to imaging centers. The patient must call, schedule an appointment, and potentially travel across town, many times waiting weeks or months to be seen and take time off work. As a result, patient adherence to ultrasound is as low as 9%. A blood test can address all of these issues of convenience, price, and accuracy. Additionally, there is a very high incidence rate in the high-risk population for liver cancer. It is much higher than that of colon cancer or breast cancer. It is about 2 to 3%. And the rate of liver cancer doubling time is very quick, about 110 days. So the screening recommendation is very frequent, and patients need to be tested every 6 months. Next, detecting HCC early is clinically meaningful. If you can detect liver cancer when it's just one nodule and the tumor is small enough, then you can do a resection, which is the gold standard, and has a very, very good outcome. If the cancer is not resectable, it may still be ablatable if it hasn't metastasized to other organs. But if the liver cancer reaches a late stage and has metastasized, then prognosis is very poor. Lastly, there's minimal competition in liver cancer. Most other liquid biopsy companies in our space are focused on colon cancer and lung cancer in terms of detection. The HelioLiver test is blood-based. It is more sensitive than ultrasound, as we've demonstrated from our trial. It addresses many of the concerns mentioned before. And our test leverages advanced multi-omics data and ML and AI in the algorithm. The HelioLiver test offers superior sensitivity for early stage liver cancer when preferred treatment options are available. And our test is currently the only liver cancer test that we know of that has been validated in a large-scale US prospective trial. So this is a comparison of ultrasound and HelioLiver. Ultrasound, being imaging-based, has difficulty detecting early-stage small lesions. You're looking at a cirrhotic liver that has lots of lumps and bumps on it. It's very difficult to determine if one of those bumps is malignant. Additionally, it's less effective in patients with high BMI, which is ironic because most patients who are high risk for liver cancer have a high BMI, with fatty liver disease being the largest contributor to high risk for liver cancer. Additionally, ultrasound requires specialized training. It requires a sonographer, it requires expensive equipment, and it requires a radiologist. And the performance of ultrasound will depend on the experience of the institution and of the healthcare providers involved. The HelioLiver test can detect very early lesions, even when the patients have high BMI, and it is easily accessible with a standard blood draw, and it can be done at any patient service center or clinic with a phlebotomist. We are also honored to have Dr. Bob Langer serve on our scientific advisory board. Dr. Langer is the founder of Moderna, as well as 40 other companies. He has created over $220 billion of aggregate value in his companies measured at peak valuation, and his companies have raised over $9 billion. And Bob has been a big proponent of our tests and our data and our science. In a study of 1,873 HCC patients, adherence to ultrasound was extremely poor. It was less than 10%. If you search literature, sometimes you'll see 20, 21% at the very highest. It must be performed in a specialized imaging center. You need proper equipment and staff. If you see the picture of the ultrasound buttons here, it's actually quite complicated. So there's been a large shortage of sonographers as demand for ultrasounds outpaces the practitioners the technicians who can provide ultrasound. And as a result, especially in areas with high incidence and low access to ultrasound, you're seeing very low adherence to testing. With a blood-based test like HelioLiver, many key opinion leaders, such as Dr. Mindie Nguyen, who you'll hear from in a bit, believe that adherence can be significantly improved with the increased access and convenience. Again, detecting early-stage liver cancer is clinically meaningful. If the cancer is detected at an early stage, the 5-year survival rate is over 70%. Patients detected at this stage also have a multitude of less invasive and more efficacious treatments available to them. The gold standard is, again, resection, where the surgeon will remove part of the liver, including the entire tumor, or an ablation can be done if a resection is not an option. If the liver cancer is detected in late stage and the tumor has grown too large, which many cases is the case with ultrasound, then the median survival rate is less than 1.5 years, and the patient is typically looking at chemotherapy, immune therapy, or late-stage drugs that are just incrementally or marginally improving lifespan. To our knowledge, we are the first and only company to submit a liver cancer liquid biopsy test to the FDA for a PMA label for liver cancer detection. Currently, we have no other competitors that we know of that are running studies intended for the FDA. Our science is based on various platforms that we have developed throughout the years, starting from UC San Diego and further being refined to practice in our own laboratory, including Eclipse, which is our wet lab platform, a fully automated laboratory platform that allows for the sensitive detection of methylation and other epigenetic features. MESA is our bioinformatics platform. We just published a paper in Genome Medicine. This allows us to combine multiple epigenetic features into a single assay and single algorithm to look at the cancer in a multidimensional view and increase our predictability. Let's talk about the TAM. So our primary market is liver cirrhosis with severe hepatitis B, which is under the recommendations by the American Association Study for Liver Disease for testing every 6 months. Adjusting for overlap, we estimate this to be 1.5 to 2.4 million patients who need to be screened every 6 months. That equals about close to a $3 billion TAM for our primary market. For our secondary market, we are targeting patients with NASH, which is a severe form of fatty liver disease. NASH affects 16 million Americans in the US. Fatty liver disease is the largest contributor to both of these markets, both NASH and the high-risk liver cirrhosis category. NAFLD, or fatty liver disease, affects over 80 million Americans, representing close to 25% of all adults in the US. And actually, that proportion holds true for the world as well. Our pipeline consists of other indications within liver cancer. While the company owns IP on a multitude of different cancer types, we are focused on liver cancer for our next 2 products. We are currently building a minimum residual disease product as well as a treatment guidance product. One thing we've realized is that with all the new immunotherapies and checkpoint inhibitor therapies coming to market in HCC, there is a dire need for biomarkers to help guide precision oncology. But for HelioLiver, we have submitted that for FDA approval. We expect potential FDA approval in Q2 of 2025. And we are also working on HelioLiver 2.0, which is an advanced version of the existing HelioLiver test to continue to stay at the forefront of competition and to provide the best possible product to our patients. Our multimodal, multi-omics computational platform can be leveraged to address the entire cancer continuum from screening, treatment monitoring, minimum residual disease, recurrence, and other cancer types. So years of work was put into the platform and establishing a data lead from which we can draw, and we have a very robust product pipeline as a result. We have an extremely talented and experienced multidisciplinary team. I'll just highlight a few people here. Gary Frazier was a hospital executive from the Dignity Health System. He's leading our growth and scale efforts as Chief Growth Officer. And he's having conversations with many health plans, integrated delivery networks that are very excited for the rollout of the HelioLiver test. Dr. David Taggart on Zoom is our Chief Scientific Officer. He holds various patents for liver cancer drugs as well as liver cancer biomarkers. And he's leading our efforts in cfDNA methylation in the lab. Dr. Shivani Mahajan is our head of computational science. She was actually one of the first employees at Freenome before she joined us to head up our computational science team. Dr. Richard Van Etten is our Chief Medical Advisor. He is a very well-respected oncologist. He is also a leading cancer expert and the director of NCI-designated UCI Cancer Center. And Dr. Jinji Hu is our Chief Regulatory Advisor. She is the former FDA lead review chair for in vitro diagnostics. Just to mention a couple people on our board, we have Chris DeRosa, who is the president of Cigna. We have Dr. Shu Lee, our founder, who has had 4 successful exits. We have RF Nakuda, who is the former CFO of Amazon, Singapore. And we have Dr. Peony Yu, who is the Chief Medical Officer of Apollomics, just to name a few. So we're going to shift gears now, and we're going to talk a little bit about CLiMB. As I mentioned before, we just got back from EASL, which is the International Liver Conference, happened in Milan this year. just last week, and we had a main stage presentation on the CLiMB trial data. There were several hundred people there physically in the audience and many more dialed in online. This is the same data that we submitted to the FDA and that we'll be submitting for manuscript very soon. In the trial, we passed our pre-specified co-primary and secondary endpoints. This was a almost 2,000 patient trial where patients were enrolled over 3.5 years, fully prospectively making this the largest completed trial of its kind. It was blinded by an FDA audited CRO, and close to 50 hospitals are represented around the US in this trial, including Stanford, who was the lead investigator, which Dr. Mindie Nguyen was the principal investigator for. Based on the very positive results, we have submitted this to the FDA, and we're looking for approval as a Class 3 medical device, and we'll be rolling out our LDT this year as well. So I'll now hand it over to Dr. Mindie Nguyen. She's one of the most respected medical authorities in hepatology and gastroenterology, liver disease, liver cancer. Dr. Nguyen is a professor of medicine at Stanford. She's also an active hepatologist. And Dr. Nguyen, if you could please review the CLiMB data with us.
Thank you, Jason. So this is our prospective multi-center blinded trial that Jason alluded to. So this trial enrolled patients with cirrhosis, which is a very established at-risk population for HCC and have been recommended for the last 20 years by all major professional societies guidelines to undergo regular HCC surveillance. And the current surveillance techniques is ultrasound plus minus alpha fetoprotein every 6 months. So this trial compare head-to-head HelioLiver test with ultrasound alone, and AFP was also done in every patient. So all cirrhotic patients who sign up get the HelioLiver test, ultrasound liver, and serum AFP. All of them also undergo multiphasic MRI with contrast, and all of these are done at about the same time with a short window at enrollment time. With MRI and ultrasound also read centrally and blinded fashion, and the patients would be classified by LRAD scores for MRI. So the not categorizable would not be evaluable or included in the final analysis here. If they are LRAD 1 or 2, they are considered as no HCC group. If they are LRAD 4 or 5, then they would be considered HCC positive. The M categories are the one that is likely malignancy but not categorizable, so they would undergo Stanford follow-up care, usually biopsy at the local sites. LRAD 3 patients would be considered indeterminate, and these patients, all of these also get the Helio MRI ultrasound at baseline like everyone else, but they will undergo serial follow-up at 3 to 6 months. So the study recommends 6 months. Some patients get 3 to 6 months according to the local PI practice. And with this, they will subsequently assign as non-HCC if they are, they don't have LRAD 5, and LRAD 5 is HCC positive patient, and the same as the M malignancy would also be categorizable as a standard of care follow-up and not evaluable here. Next slide, please. So this is our trial that includes for the validation cohort. The initial cohort was in the derivation cohort. And for the validation cohort, we have 1,222 negative HCC patients and 46 HCC patients. So the risk in the cirrhotic cohort is around 1% or so, 1 to 2%. So this is in the expected range for us to get the HCC number with our sample size calculation. So you could see that the HCC positive patients are older, and that is expected, and they are more likely to be male. The ethnicity and racial spread, as you could see here, we have close to a 50-50 for Hispanic and non-Hispanic spread among the HCC. more non-Hispanic in the HCC negative group. Obesity rate is about 55-56% in both HCC positive as well as non-HCC groups, which fairly reflects the population prevalence of obesity in our country for this age group. Diabetes close to 50%, and the strength of the study here is that the 52%, 82% of community-based center enrollment of this study. So the results are hopefully more generalizable to the overall population of people with cirrhosis rather than just patients at referral tertiary care center, which is fairly typical of many trials. For liver etiology, the majority or half of the group here is NAFLD, so fatty liver, and now there is a new name for it called MASLD with an M for metabolic associated steatotic liver disease, but they are fairly similar in terms of the prevalence in the population. And then we have a good spread of viral and alcohol associated liver disease. Next, please. So a review here of our flow, patient flow chart. So 1968 participants total. As I mentioned earlier, 412 enrolled before June 2020, and they were assigned to the training cohort, and then the 1556 assigned to the validation cohort. Of these, the evaluable cohort is 1285. We lost 271 patients due to variable reasons. 200 fail the screening criteria, and 16 of them, the ultrasound or MRI, not evaluable. A lot of it due to reasons that the disc could not be de-identified and sent to central reading. And 6, 30, and 19 due to specimen issues. And of the 1285 evaluable at initial visit here, we had 45 HCC indeterminate at initial visit, 17 of those lost to follow-up, and at the end we have 46 HCC, 4 at follow-up visit HCC, and 42 at the initial visit. And we have 1222 non-HCC patients total. Next, please. So here is the spread of our HCC cases, 46 total. As you could see here, the majority of the patients had early stage, 65%, only one tumor. 8.7% even had lesion less than 1 centimeter, 37% 1 to 2 centimeter, and 28% 2 to 3 centimeter. So total, we only had 26% with lesion 3 centimeter or larger. And when we consider the T stage, the similar finding is seen here, 58% T1. So the cohort probably reflect a surveillance cohort with the majority of the patients here diagnosed at pretty early stage, from early to very early stage. And this despite the fact that 82% of these patients come from community-based center. Next, please. And this slide demonstrates the details of how the study met both the primary and secondary endpoints here. So the primary endpoint for sensitivity difference here is to detect a 5% superiority margin. And as you could see here, both the primary as well as secondary lesion less than 4 centimeter and exploratory lesions less than 2 centimeter here all met the 5% superiority margin. And we also met the 10% non-inferiority margin for specificity of all the primary evaluable outcomes. Next, please. This is our main results. As you could see, the first bar that is in darker shade is for sensitivity, and the lighter shade is for specificity. So comparing the HelioLiver test alone with ultrasound alone, which is the primary comparison here, 48% sensitivity for the HelioLiver test compared to 28% for ultrasound alone. And this is for all lesions together. And for specificity, 88% versus 94%. When you look at AFP alone, we just put it up here as 22% and 98% for reference, but this is AFP alone is not part of any of the guidelines for HCC surveillance at this time. So ultrasound plus AFP, and this one, the sensitivity is 35%, and specificity is 92%. And the cutoff of AFP here is 20, is routinely done in research studies of this topic. But the true high abnormal for AFP is usually about 6. And at this level, sensitivity may be higher, but specificity would be very poor. Next, please. And this is the result summary again. So Helio test alone 48% sensitivity and ultrasound alone 28%. And if you look at the Helio test and ultrasound 52%. So it did not really add to the sensitivity of the Helio diagnosis test if we add ultrasound. So in here we have the Helio liver test, 20% more sensitive and 6% less specific than ultrasound alone. HelioLiver test also had a higher sensitivity than the combination of ultrasound and AFP. Subsequently, I would like to show the results where the test really separates itself here, and when it really matters in a clinical setting, which is the diagnosis of early HCC. So if we diagnose large HCC better or the same, it doesn't really change the outcome of the patients in terms of survival very much. But if we can diagnose the patient with earlier tumor stage, these patients can potentially get curative treatment where the survival at 5 years would be 70–80% versus 10% or less otherwise. So in here, the first graph you can see here is to look at the subgroup analysis by T-score. So as you could see here, if we look at T1, then the difference in the sensitivity between the HelioLiver test and ultrasound is fairly large. In T2 to T4, there do not appear to be significant differences here. Now, looking at the largest HCC lesion size, and if we use 2 centimeters, because this is usually the cutoff clinically when we start feeling that the lesion is small versus large. So you could see that the HelioLiver test here could detect about 25% of lesions less than 2 centimeters, 2 centimeters or less, when the ultrasound did not detect any. And again, these ultrasound are all centrally read and are all in blinded fashion. The difference does not seem to be significant here when you look at lesions more than 2 centimeters. Next, please. And by etiology in subgroup analysis, because we have 4 major groups here, so the sample size in each of these are pretty small, and it's hard to tell in here if there are significant difference or not. Numerically, the sensitivity for Helio test is more than ultrasound for NAFOD, but again, none of these comparisons are statistically significant. Next, please. Same here for race and subgroup analysis by race between white and non-white. We see that there may be a numerically higher sensitivity for both white and non-white here between HelioLiver test and ultrasound, but sample sizes are small, and there was no statistically significant difference. Next, please. Same thing for ethnicity, Hispanic and non-Hispanic here. Next, please. So in conclusion here, the CLiMB trial met the co-primary endpoints for superior sensitivity 5% margin and non-inferiority inferiority for specificity, 10% margin, compared to abdominal ultrasound for both the co-primary endpoints and the secondary and exploratory analysis endpoints for sensitivity. The HelioLiver diagnostic test was more sensitive than ultrasound for detection of early HCC, particularly 2 centimeter or less. So this offers an alternative for accurate and easy to use blood tests for the detection of HCC, and early HCC detection has been shown to improve, to significantly increase the treatment choices, especially curative treatment, and vastly changes the long-term survival of patients with HCC. So, as mentioned earlier already, the HelioLiver test results here and as studied here as a blood test by itself, not in combination with ultrasound. And the ultrasound in clinical practice, especially since the pandemic, the access has been much poorer. At Stanford, average wait for routine ultrasound for my patients is 5 months, and that has really been a barrier. And I will end here and will be glad to take any questions.
All right, I'd like to invite the audience to ask questions. Those that are on the webcast, there should be an option for asking questions, and we'll get that. So go ahead, Vijay.
Great. Dr. Nguyen, thanks for your comments. I have a few on the trial. What's the current patient workflow? What's the referral channel? When I look at the trial enrollment period, 3.5 years for 3,000 patients, was that like a long enrollment period in mind of how does the patient workflow in this thing?
Mindie, did you get that?
I'm sorry, I couldn't hear anything at all.
So the question is around the patient flow in the trial. 2,000 patients over 3.5 years. Is that fast or is that slow? And maybe review the clinical trial design one more time.
Okay. So the patients need to have cirrhosis according to established criteria, and all patients would receive blood tests for alpha-fetoprotein and the HelioLiver test. All patients also got liver ultrasound and also multiphasic MRI with contrast. All of these are done at the initial enrollment with a short window, as I mentioned earlier. And all of these patients would have their ultrasound and the MRI images sent for central reading, and all of them would end their follow-up at that time unless they have LRAD3. Then they would get subsequent imaging to determine their up to 18 months to determine their status as HCC or non-HCC. And I acknowledge the audience mentioned that this is the trial took a couple of years to enroll here. But I want to also mention that this was started like around during the pandemic. So at least at our center, everything was slower in terms of getting started. And also patients were not coming in to the clinic for anything due to various reasons, access at clinic as well as fear of COVID, etc. So it did not pick up until later on, at least for us and likely other centers as well. And the other thing is that the cirrhosis, there are very specific criteria, and the screen fail, as you could see in here, 200 of them did not quite meet everything, and even this is after screening by the PI already. So some of these may explain some of the speed of the enrollment.
Maybe one more for you.
Wait, you got a microphone so she can hear this time.
Yeah, thank you.
Hopefully you can hear me now.
Yeah, I
Dr.
can.
Nguyen. Perfect. So what was the no-call rate for the blood test? And I'm curious how that compares to, you did mention the 9% adherence rate for ultrasound rate. Maybe talk about the test performance.
Yeah, so so this study did not was not designed to address the ultrasound adherence, as in HCC surveillance, which is a surveillance of ultrasound every 6 months, and the 9% adherence, that is data that my group at Stanford performed in 80,000 patients with cirrhosis cohort from a real-world insurance claims database. So I believe the 9% adherence is probably reflect the real world. And this database that we use went up to early 2021. So it's pretty fairly recent data. So in this study, the adherence in terms of the percent of people who come in to get ultrasound is much higher, of course, more than 90% here. In here, you could see that only 157 did not undergo ultrasound and or MRI. And some of this is because they changed their mind. They didn't want to do it anymore. But a lot of time due to scheduling issues, and as I alluded to, despite the much effort to coordinate with radiology center, we were not able to schedule them during the window of the study. So in here you can see 200 of these. So close about 10% or so. So 90% came and get both the ultrasound and the MRI for the study.
Gotcha. And maybe one more from me.
Mm-hmm.
Anything on the baseline demographics that stand out for you? I think you mentioned 50% of patients were NAFLD, 50% obese, 35% viral etiology. Do those numbers match what you would expect in a general patient, the background prevalence in the population setting?
Yeah. So this is a cirrhotic cohort. So among the cirrhosis for the contemporary, if you read older papers, you would find that viral hepatitis may be more common, but we have made great advances therapeutically with both Hep B and Hep C. So we have a very good and effective treatment for hepatitis B since 1999. And we have very good curative treatment, like 98% cure rate for hepatitis C since 2014. So because of that, in the last 5 to 10 years, the percent of people with end-stage liver disease, like cirrhosis or liver cancer, due to viral hepatitis, have really gone down. And the major cause of cirrhosis and HCC now is alcohol associated, as well as non-alcoholic metabolic associated liver disease. So I think that our spread here, about 50% NAFLD, really reflects the more contemporary makeup of the cirrhosis populations in the US.
That's helpful, Dr. Nguyen. And maybe last one, if you had to summarize everything, right? I mean, for us, the data looks very, very good, right? It's you hit the superiority on sensitivity. Even when we try to look at the numerical values by different settings or tumor size, the blood test came out better than ultrasound. How would you characterize these results? Is this like practice changing for you? Maybe just give us some qualitative comments on your opinion of the test. Thank you.
Thank you. Yeah, I think that the difference of 20% overall for all sizes, come together is significant enough that it would change our practice. And this is but this is only one part of the equation for in clinical practice, because in clinical practice, we consider effectiveness of the test, not just efficacy. So in clinical trial, here is the result we see in terms of efficacy. But effectiveness also depends on the adherence rate. So if the patient's adherence is 100%, then effectiveness and efficacy would be the same. But life is never like that. So the adherence rate for a blood test is most likely much better than with an ultrasound. And the adherence to ultrasound may differ according to setting. So let's say if you have a practice that people can come in for an ultrasound anytime, then maybe you'll get much better adherence. But in the vast majority of cases, ultrasound require advanced scheduling because the patient need to fast for at least 4 to 6 hours, depending on the center. So it's never going to be a really walk-in test. That's one. And second, due to the shortages of the experience center as well as sonographers, especially since the pandemic, the access to ultrasound has been really, really poor, even at a center like our center. And so there are system issues that make the adherence rate poorer. And there's patient factor. People have to call and schedule. They have to schedule in a day that they can fast. They can come in. They have to spend a couple of hours for the test on the day of the test, whereas blood test usually is always easier to convince a patient go get blood test. So, and most patients at this age group especially are used to see their primary care doctor and getting blood tests at least once a year. So I think that together with the efficacy and the adherence and barriers to the test itself, I think that it would be much easier on the patients as well as for clinicians too, because it's quite stressful for the clinician if we order a test and we can't get the test for 5 months, and during that time, what if there's something bad in there that we are missing? So so I think that it would be very helpful. Now the other thing is that for small lesion, as you could see in here, ultrasound detected 0 of the less than of the 2 centimeter or less lesions, and this also increased the confidence for both patients and doctors, and that together may help with the adherence as well.
Great questions. Go ahead.
Okay.
Yes.
Yeah, I have a couple of questions here. One, I understand that this is done for people that have cirrhosis, but do you see in the future, as things move forward, that you'll be able to get a mass screening out of this in the manner that PSA is done for prostate cancer? Next question, I was unsure, is this test done in the facility where the blood is drawn, or it's required to be sent out to a lab? And third, going forward, do you have in the pipeline other ways of improving the test to improve the sensitivity?
Thank you. So for the first question, mass screening for liver cancer is not currently recommended, and I don't expect it to ever be recommended like the way you do for prostate cancer or cervical cancer, for example. So HCC is a very bad cancer, but fortunately, it has a pretty well-defined at-risk population. And the one at-risk population that is not disputable is cirrhosis. And that's why the trial was designed to include only cirrhotic patients here. There are 2 other groups of patients that are also at higher risk for cancer, but the screening or surveillance criteria vary a little bit among the different guidelines. So one is hepatitis B. So many guidelines would recommend liver cancer surveillance for all hepatitis B patients, but the US guideline currently only recommend for people older than 40 for men and older than 50 for women if they do not have cirrhosis. And that's because it is felt that the risk is too low below these age cutoffs to make it cost effective. But that is being revisited now because these were based on pretty old data or lookup data. in Asia Pacific, where the cancer is a lot more common, people do not have an age cutoff for hepatitis B non-cirrhosis patients. So in my practice, I perform HCC surveillance and screening for all my hepatitis B patients without cirrhosis, and of course, the people with cirrhosis too. The other group that still not mass screening, but you may say it is because 35–40% of the US populations and globally have the disease, which is NAFLD or MASS LD, fatty liver disease. So it is very well reported that fatty liver disease patients without cirrhosis is also at risk for liver cancer, just like hepatitis B. However, again, the risk is low in the young population, so the people who have no fibrosis at all. So guidelines have been kind of inconsistent in what to recommend. But many guidelines now at least will also include patients with stage 3 fibrosis. So not just a stage 4 cirrhosis patient, but also stage 3. So these are evolving area, and I expect that in the next 3 to 5 years we will have no more, and the criteria can be better defined.
I'll answer one of the questions, the pipeline for improving the product. We do have HelioLiver 2.0, which we're currently working on. The way the FDA works, you can't really have continuous improvements without locking down the assay or the algorithm. So it has to be a stepwise introduction of new products. But we plan to release results on HelioLiver 2 in the future to continue to stay ahead of competition and provide the best products to patients and physicians. But right now, the data is very good because you have to consider that the trial is 60% stage 1 cancers. So it's a very early population.
And his last question about in a lab, it has to go to an actual lab versus being in the practice.
Yeah, so it's drawn at the site, so at the hospitals and clinics, and then the blood is sent back to our laboratory in West Lafayette, Indiana. We have a central laboratory. Thank you for the questions. Mindie, I have a question from you, from the web chat. What is the specificity of the test for early stage HCC 4-centimeter lesions or smaller?
Okay, so let me pull that.
So it should be the same because we're looking at the negative patients.
Yeah, so it's so the specificity for the smaller lesions, it's pretty similar. They all, like, you can see the slight difference, 88%, 92%, but they all around 90% for specificity. And I wanted to mention that, from the clinical perspectives of all these, what I think is more important from the patient and provider perspective here is a sensitivity, because we do not want to miss a cancer. So the cost of missing a cancer is a lot more. And of course, we don't want specificity that is only 50 or 60%, but the difference between 88, 90, 92, 96% actually not that large to be clinically significant.
Thanks, Mindie. I have another question, but it's for the management team. So do we have any more questions for Mindie? Yes.
Hi, Mindie. Thanks for the nice presentation and explaining about sensitivity more than specificity. One quick question, I guess, Howard, when he was mentioning about lab, I guess he had Theranos back in his mind. But anyways, I would like to know regarding HCC, what is the possibility that these patients, I mean, these subjects that were the test subjects, what was the COVID-19, I mean, antigen or the COVID-19 virus impact on some of these patients during the test period, I would say? The virus impact and all. Was that studied as part of this diagnosis?
If I understand correctly, is your question regarding like how many % of these patients had COVID and how that may have affected the test?
Yes.
Yeah,
Because there might be a variance there, right?
Yeah. am not totally sure if we collected this data. So I don't, David Taggart, our Chief Medical Officer, I don't know if he has the information. However, I doubt that that would be a significant number, if any, because if they have COVID, they would not be allowed to come in to the medical center. At least, for our center. Yeah, so most likely they exclude it. And one of our investigators here, Dr. Van Etten, feels that they will exclude it if they have active COVID, and that would be my understanding too, because they would not be able to come to the medical center anyway, regardless of the participation of this study.
And again, the antigen, would it block those initial tests? Would it cause any interruption in that, or like receptors if they are taken the antigens?
You
the
mean
tests
the
for
COVID?
PLC and HCC? Yeah.
Yeah. So I am not aware, but I don't know if it's really relevant for this cohort, because I don't believe that we included anyone with active COVID.
Yeah, it's not something that we studied. It would be purely speculation, but we have done extensive analytical bench testing studies for the FDA with interfering substances. I don't believe COVID was one of them, but the test stands up from an analytical standpoint. Any other questions for Mindie? Let me see here. Okay, maybe I'll invite the management team. Oh, go ahead, Rick. You can unmute.
I would just, yeah, this is Dr. Van Etten from UC Irvine. I would just say there's no reason in my mind to think that any COVID antigen, either from the virus or from a vaccine, like the spike protein, would affect either the methylation pattern or the protein biomarkers that are measured in HelioLiver.
Yeah, I would agree.
Thanks, Rick. Okay, Gary and Alex, if you could please join, and then we'll open it up. Mindie, I know you have to drop off at 8 Pacific, so feel free to drop off. If anybody has additional questions, email us and we'll send them to Mindie.
Thank you.
But we'd like to open it up for management Q&A. So anything is fair game here, financing, commercialization. Please, Vijay.
So I guess my first question is the referral pathway here. When you think about building the commercial organization, what is needed to happen here? Do we need guidelines? These patients, are they seen by targeted, liver clinics? Like how many clinics are there? What's the sales force needed to hit the target audience?
You keep the microphone. So the pathway does not require that much of it requires training. So it requires practice training, but they're seen by not only hepatology centers. If you can imagine, the general population is most likely seen in a primary care clinic, most likely seen in the community setting. The good news per Mindie's presentation is that they are very identifiable. And so the ability, unlike the general population where you should be doing direct-to-consumer marketing and things like that for every male over 45 if it's prostate or something like that, for this one, anyone diagnosed with cirrhosis, for example, right? You should be able to go into an EMR database, and if you are an integrated delivery network or a managed care organization or something like that, you can identify these patients right away. So it makes business development and sales easier, for lack of a better way to put it, because you can follow the population that way. Same thing with hepatitis B and everything else. So you're talking a smaller engine, if you will, sales and growth engine, than one that is targeting the whole population. You're talking a smaller marketing engine that's required, and a really well-focused approach to going after them, because now you're saying, well, let's go to the folks that have covered lives, right? That match the population that we're looking for. I will say this last thing is that, if you look at the population that are NAFLD, and have high BMI and are suffering. And they tend to trend low income. They trend older, so over 65. They trend Black, Indigenous, BIPOC, if you will. And so just putting on a health administrator's hat, that's Medicaid, right? That's Medicare. And so now you're saying, okay, well, who's paying for these patients? Well, you got managed Medicaid, managed Medicare, you got Medicare Advantage. And so now you start to really hone in on the organizations that you're looking for. Does that answer your question?
It does. It does. And maybe related to that, Justin, for you, what should be the revenue ramp for a test like this? Assuming we have broad coverage reimbursement, it's part of the guidelines. Is this 50, 100, 150 by year 3?
It's significant because we would be the only product in the market. The only analog that we would draw would be to Exact Sciences back in 2014 when they were FDA approved. I don't think in this meeting we can discuss any forward predictions, but we're happy to take it offline and try to answer that question better. We think we can get a significant share of the TAM.
Understood. And maybe last one here, maybe for Dr. Van Etten. Did you, when you ran the trial, did you ask these enrollees on their willingness to take a blood test versus ultrasound, or do you have any data, and would you have any follow-up health economic kind of studies?
Well, what I would say is the trial was done at centers that were actively carrying out surveillance for HCC, and we intentionally selected centers that were actively screening patients versus new patients coming in for screening, because in that latter population, you're going to catch predominantly people that have larger tumors because they haven't undergone active surveillance. So that the trial was designed that way, and it worked out exactly that way. These patients, I think, as Dr. Nguyen said, are getting blood tests all the time because of their underlying condition. If it's Hep B, they're getting screened for viral loads. If it's people with cirrhosis, they're being tested for liver function tests and kidney function tests all the time. So the blood tests are part of the normal standard of care that they're used to. So I think only real world experience will allow us to see what the penetration of the HelioLiver test is once it is approved, as we anticipate it will be. But I think that it's entirely plausible that the adherence is going to be very, very high. Did that answer your question? There was another part to it.
There was another part, and yes, it did answer on any follow-up health economic studies when you
yes.
reach out.
So interestingly, Dr. Nguyen is designing such a study right now. We predict, just based on some pretty simple calculations, that the early diagnosis of hepatocellular carcinoma and the ability to apply potentially curative therapies, therapeutic modalities, will in the long run positively impact healthcare costs. And that's because the people that have larger liver disease, they have poor long-term survival, but over that next 5 years, they consume a great amount of medical care. They go into advanced liver failure. They are getting, for example, targeted therapies such as kinase inhibitors, which are purely palliative in nature. And it's our expectation that that's going to prove to be the case. And so we're going to actually launch such a study to try to calculate what the healthcare benefits would be of early diagnosis.
Great, thank you.
I just want to add follow up on what Dr. Van Etten mentioned about blood tests. So for cirrhosis patient, guidelines recommend that they need to get clinical assessment and blood tests every 3 to 6 months. So HCC surveillance is ultrasound currently every 6 months. And so they would be showing up and getting blood tests. And when I looked at the TrueVan database recently, which includes 253 million US enrollees with private commercial insurance. The percent of cirrhosis patients getting blood tests, any blood test for any doctors, is about 80% each year.
Great, thank you.
Rupesh Kumar here. Gary mentioned a very important point. This is really a gift for the low-income families for sure. That being said, what might be, just wondering, what might be the out-of-pocket cost for this? I mean, would it be same as a typical blood test?
Yeah, I mean,
I mean,
I can only
projections.
speak for the plans that we've seen that have adopted it as coverage with prior authorization. So if it's Medicaid, it's generally nothing out of pocket.
Yeah.
Because that's the kind of the point of Medicaid, right?
That's-
For Medicare and Medicare Advantage, similarly, right? You know, you're already on a Medicare Advantage plan. It's when you get into the PPO and the commercial plan space where plan by plan, each of us have a different plan, and that determines our, you know, our premium, our co-pays, our co-insurance. So, you know, the Medicare rate is where we start. So that's what will be essentially charged. How much your plan covers, 90%, 50%, that'll be, you know, determined based on your plan. But for the larger populations that we're targeting, there should be zero out of pocket because they're Medicaid and Medicare predominantly.
Thanks.
I have one from the audience. Can you describe the development of the HelioLiver DX test and how AI is incorporated? Shivani, do you want to take that one?
Sure, yes. The development of the test starts from discovery. So the first step is obviously to identify the most informative and predictive biomarkers. So to do that, we first started off with tissue. So using cirrhotic tissue and HCC tissue to identify the first set of biomarkers. So essentially casting a wide net at first, and then sort of narrowing it down using further plasma from HCC and cirrhotic patients to finally build our targeted panel. The test building process was actually also iterative, where we want to avoid overfitting. We want to avoid what we essentially call train test leak. So we kept the sets of samples that we did discovery on different from the samples that we actually did training on when we were evaluating model performance, and finally only rolled in those patients for model training once we had evaluated their performance on samples that were previously unseen. So starting with case control data, we then evaluated the model on prospective samples, and finally for the final model, trained model on both case control and prospective samples before it was validated on the CLiMB study. The algorithm that we use essentially uses information coming from cell-free DNA methylation markers, also protein biomarkers, as well as patient demographic information. All of that is combined using advanced AI algorithms that we have customized and built in-house, which I'm happy to provide more details in. You can follow up in an email, or I can answer further follow-up questions.
Thank you, Shivani. I have another one here. Can you provide an overview of the Eclipse lab platform and the MESA sequencing assay? Dave?
Yep, I'll take that one, Justin. Thank you. The Helio Eclipse platform consists of automated and optimized methods for both cell-free DNA extraction, generation of next-generation sequencing libraries, and an enzymatic conversion step. Also target capture, so we're pulling down just our targets of interest, and then also next-generation sequencing. Now, importantly, as compared to traditional approaches that use DNA methylation, we're using an enzymatic approach, which is far more gentle on our DNA, as opposed to the traditional bisulfite conversion approach, which is known to damage DNA. And because we aren't damaging our DNA in the same way, we're then able to use our MESA approach, which is to evaluate multiple epigenetic features of our sequences simultaneously, including DNA methylation, nucleosome organization features, fragmentation features. And in the manuscript that we put out earlier this year in 2024 with Dr. Wei Li's group at the University of California, Irvine, we can show that by putting all of these features together within a single test, then we can have test performance characteristics that are superior to tests that are just looking at one feature.
Thanks, Dave. Can you provide insight on the general advantages of contemporaneous MRI, as well as in the context of the CLiMB study? I'll go ahead and answer that one. So The benefit of having contemporaneous MRI in our study is that it is fully prospective. In a lot of other studies, they're case controlled. So you're enrolling HCC patients after they've already been diagnosed. So in practice, that means the patient already got a positive ultrasound and a positive MRI. They could have had HCC 6, 12, or 18 months ago, and the ultrasound didn't catch it, right? So you're going to have a lot of bias in the data set, and ultrasound sensitivity is going to look a lot higher than it is in the real world. So CLiMB had contemporaneous MRI. MRI was provided for all patients, and it is probably the best measure of the true sensitivity of ultrasound for early stage cancer. Let's see. How do you account for the relatively large standard error bars when looking at the test sensitivity by HCC lesion T-score and size, as well as the subgroup analysis for etiology of liver cirrhosis? Dave, do you want to take that one?
Yeah, I'll take that one. So as we take our patient population, we try and break it down, right, into more and more categories. Then you have fewer patients within each of those categories. And as you start having fewer patients per category, then the 95% confidence intervals are expected to get larger.
Thanks.
And I would emphasize that the study wasn't powered necessarily to detect significant differences across those different subsets.
That's right. I have another question here. Have you received any feedback from the FDA on the PMA application? If not, when do you anticipate hearing from them? Dave?
Sure. We expect the majority of questions come up from FDA about 100 days after submission, which would put that around August to September.
Okay, one more question. Plan on generating revenue, major business plan after IPO. So I'll just say we can't comment on an IPO, but Alex or Gary, why don't you take the revenue question?
Yeah, I think the next round of fundraising obviously is going to focus primarily on commercialization, both sort of boots on the ground, like Gary was talking about, but then also scaling the lab capacity in West Lafayette, Indiana. Right now we're at a capacity that's in sort of the tens of thousands, and we want to scale that facility first. So primarily commercialization, capacity, and then the pipeline work that Justin showed earlier on.
Any next steps on the CLiMB study? Gary?
Next steps on the CLiMB
Yeah.
study? Well, I mean, in as much as we can dovetail into commercialization and capture longitudinal data, real world data, that's the goal ultimately. And so it's also the reason why we're partnering with managed care organizations specifically to start, because it's more of a B2B approach versus, again, a D2C approach. And so by targeting those organizations that have economic incentive and clinical incentive, they typically employ the doctors as well. We can also partner with them on the data capture part of this thing, right? And, a year, 18 months, 24 months down the line, we'll be simultaneously capturing that clinical data. And that actually is what feeds the flywheel. So the more efficacy and effectiveness data that you can capture from the real world, the more utilization you're going to see in the real world. And then that, of course, feeds into more data, and all of that feeds into more revenue. And, that's the flywheel effect of this whole thing. Hopefully that answered the question.
Any other questions from the audience?
There's one right here.
Please.
Yeah, a little bit more on the business side. Can you comment on what your last round of funding was and what the valuation is? How far that takes you down the road? Then I'd like to know about management ownership and institutional following.
Go ahead, Alex. Why don't you take that one first?
Yeah, so the last round of fundraising was in 2023, and we raised at an effective valuation of around 220 million.
Yeah, and then we've got runway through the rest of the year, and we will be getting ready for an institutional fundraising in the very near future. Is that answering your question? Okay, thank you. Additional questions? Let me see if anybody. I think that's it, unless anyone has any other questions or. Okay, well, I'd like to thank the audience here in person. I'd like to thank the audience on the webcast. I'd like to thank Dr. Mindie Nguyen, Dr. Richard Van Etten for signing on. Also like to thank NASDAQ for providing the facility. So appreciate everyone's time. Thank you.